Assalamualaikum and good morning class. Here is a short video on demonstrating how do you conduct a food test according to O-level standards. So the main foods that are tested on for food tests are starch, presence of starch using iodine solutions. So it's iodine solution. Iodine solution is yellow-brown in color, and in the presence of starch it turns from yellow or brown to blue-black. In the absence of starch, then you would write that iodine solution remains brown or remains yellow in color, or the color remains yellow. Then we also test for reducing sugar. Now in the past you may have learned it as test for glucose. Now this is not necessarily true, but it is a test for all reducing sugars. All of the complex and simple sugars that you have learned are all reducing sugar except for sucrose. Sucrose is not a reducing sugar, and the reagent that we use is Benedict's solution. You can see that it is blue in color. Now, in the presence of reducing sugar, after we conduct the test, this blue color will turn either yellow or orange or red, or green, if reducing sugar is present. If absent, then the color just remains blue. For tests for proteins, we have burette. So this is also a blue solution, but you can see that the color is slightly different from Benedict's solution. So the label will be clearly given to you in the test or in your practical, so make sure that you read the labels properly. Burette is used to test for proteins. In the presence of protein, this blue color turns violet or purple. The last one for the test for fats, we have then ethanol emulsion test, and the reagent we use here is 100% ethanol or 100% alcohol. The label is not here, but it will be given to you next time. So if the food test is on solid food, the food specimen given to you is for solid food, then the food material has to be cut up into very small pieces. How small? As small as possible. We'll say toothpaste consistency. If the food specimen given to you is liquid, then it's very easy. You just need to add equal volume of the reagent. So here we have a piece of potato, and this potato, if I'm asked to test 4 different food tests, then I just chop it into equal pieces here. I've already cut it up. I'll put it into a petri dish onto a side. Now the first one, if we are testing for iodine solution, this is actually quite easy. You don't have to chop it up. It's the only one where you don't have to chop it up into small pieces, even though it is a solid food specimen. So what you do is you place it on your white tile. White tile, because it's a very clear background, you can see the color change very easily. So you take your iodine solution. Iodine solution comes in a dropper. Now do not go so near that it touches your potato, or your food specimen. So it can be 1 or 2 centimeters away, and then just drop it from a distance. So just drop 1, 2, 3, and that should be enough to show you any color change if starch is present. So what do we notice here? So you can notice there is that you can see the blue-black colorations. Blue-black colorations. In the presence of starch, iodine solution has turned from yellow to blue-black. So I can say that potato contains starch. Alright, next I want to show you how small to cut your potato pieces. Now, even though you're given a lot, you don't have to use everything. If there's skin, just remove the skin. You don't really want the skin. This looks like a bit like a cooking show, so you want to chop it into very small pieces. The scalpel blade is very sharp, so make sure that you don't cut yourself. You'll notice here some students don't know that there is a blunt end and there is a sharp end to the scalpel, so you use the sharp blade to cut. Cut it into small pieces. So ideally, okay, what we want is you want to cut the food specimen into very small pieces. Of course, you need to look at the time as well. You don't want to spend 15 minutes, half an hour just cutting food. So the idea here is we want to break the food into smaller pieces to increase the surface area. That's one thing. And the other reason is we also want to break down as many cells as possible, because these nutrients are found inside the cells. So we want to break the cell wall, we want to break the cell membrane to release those nutrients so that we can test for these more easily. Next, okay, so what do we do with these chopped out food specimens? So we want to take a bit, we place it into a test tube. Here I have a boiling tube, so it's easier. It depends on what you're given in the exam. Okay. So you don't have to put everything in, just enough to get you enough nutrients. I'll put this away. And then what I have here, this is distilled water. You can use tap water as well. So what we want is, we want to add about 2 cm cube of water. Now if you look carefully, you will see that there are markings on your pipette dropper here. Oh, where is it? Okay, so around here, this is 1 cm cube. So you can adjust how much water is in your pipette before you squeeze it in. If you don't think this is enough, this is about 2 cm. Okay, you can just approximate. So I'm going to take maybe one fingertip volume of water. Okay, I'll add that in. Okay, so once you add that water, you want to shake it. The reason why you're shaking is, again, you want to dissolve any of those nutrients present in the food into the water. So what essentially you are creating is a solution that has dissolved nutrients in it. Okay, so shake it for about 1 to 2 minutes. Actually, 30 seconds is good enough, but if you're asked to write down the instructions, that you want to write 1 to 2 minutes. Okay, next. So what we realize here is we have some solution, but you also have a lot of solid food specimen at the bottom. So we don't want the solid food specimen to get into our way of observation later on. So we want to separate the liquid portion from the solid portion. This is called decanting. So very carefully, what you want to do is so this is a process of decanting. We are just pouring the liquid portion very carefully into another clean test tube or clean boiling tube. So what we have here, I have the solid material left and just liquid solution. Okay, so then we have this liquid solution, and if we are testing this for protein, okay, then I'm going to add equal volume of burette solution. You don't have to measure it accurately, you can just estimate. So look at here, this is maybe around half of my fingertip, almost one of my fingertips. So I'm just going to put my finger here. So I'm going to add the solution until it is about double of this portion. So if you're not sure, you can bring the color up for comparison. Shake it. Okay, so has the color turned from blue to purple? Hasn't changed. So we can say that there is no protein present in this food specimen. Okay, here I have some edamame beans that have been crushed up, and I have added 2 cm cube of water to the crushed beans inside a test tube, shaken it, and it has been sitting for about 5 minutes, enough time for the nutrients to dissolve into the water. And next I'm going to decant into a clean test tube. So just pouring off the liquid portion into a clean test tube. Trying not to get any solid food specimens into the test tube, but if you do get 1 or 2 pieces, don't worry about it. You just don't want the bulk of it to enter into my test tube. Okay. So test for proteins is using burette solution. You want to add about equal volume. So normally I just place my thumb about double of that I already have. Okay, to avoid contamination, I don't touch my dropper into the test tube. Okay, just hovering above and squirting. So I have about equal volume, and look at the color change. It has turned from blue to purple. Okay, this was the original color of burette. It's quite similar, but I think you can tell the difference, okay, between the blue burette solution and purple. I think that's better. Yeah. So there is presence of proteins in beans. Beans are a good source of proteins from plant sources.